Hi everyone, my name is Sydney and I'm a grad student at Southern Illinois University in Edwardsville. I'm currently working on my master's in biology and I'm focusing my thesis on genetics and microbiology. I recently gave this presentation in my recombinant DNA class. I put so much research and effort into it that I decided I wanted to share it with more than just the people in my class. I know that there are a lot of people that are really hesitant about getting the new COVID-19 vaccine. So I'm hoping that after watching this, I've answered some of your questions and maybe you're a little less hesitant about getting it. So let's get started. So really quick, just a quick outline of what I'm going to be talking about. First, we're going to go over the basics. What are vaccines? What do they do? And then we're going to talk about the conventional vaccines, the two original vaccines that were first made. And then we're going to discuss why we need recombinant vaccines, how they're made, and what they do to your body. And then we're going to see how recombinant vaccines can be improved. And then I'll address some of the claims and concerns from the anti-vaxxer community. So first, what are vaccines? What do they do? What they do to your body is they're going to be prompting your body to make antibodies. Antibodies are these blue and yellow Y-looking things that are specific to antigens. Antigens can be found on viruses, and these antibodies match them like a little puzzle piece. Once these antibodies attach to the antigen on the virus, the antibodies kind of send a signal to other molecules that are responsible for destroying viruses to come and destroy that antigen and anything connected to it. Vaccines also give you immunity to a disease before contracting it. So unlike the olden days when vaccines didn't exist, you can be immune to something without ever getting sick with it. And then of course it's preventing diseases, whereas most medicines are going to treat them after you've already been infected. So there are two conventional vaccines that I want to talk about. The two original vaccines that were first made by scientists. So the first one is live-attenuated vaccines. Attenuated just means weakened. So these vaccines are going to use a weakened version of the virus. So this virus isn't going to replicate or infect as well as it would normally inside your body, giving your immune system a better chance at killing the virus. And while you're killing the virus, this weak virus, your body is making antibodies to prepare for future infections from the same kind of virus. And currently, the live attenuated vaccines are responsible for helping measles, mumps, rubella, rotavirus, which is like a stomach virus in children, smallpox, chickenpox, and yellow fever. Another type of conventional vaccine is going to be the inactivated vaccines. Now this uses still a whole virus usually. There are non-whole virus inactivated vaccines, but for simplicity, we're going to assume that both of these use the entire virus. The inactivated vaccines use the killed version of the virus. So this virus is killed in a lab and it has no chance of coming back to life and infecting you. But because the virus is there as a whole in your body is still going to create antibodies and have an immune response and make you immune to whatever it is you're getting vaccinated for. So currently there are inactivated vaccines for polio, influenza, hepatitis A, and rabies. So if we have those two other vaccines, then why do we need recombinant vaccines? It's a valid question. Those other two vaccines come with their own issues. The live attenuated vaccine is difficult and time-consuming to develop. The measles vaccine, for example, was the first ever live attenuated vaccine, and it took 10 years for scientists to make it. These vaccines are also very high maintenance. Because it's a live virus, they have to stay in a live tissue culture when they're being made. They're also very picky about their conditions, like the temperature that they're going to be kept at. And they're not recommended for immunocompromised people. People with conditions like diabetes or cancer, or even the elderly or newborn babies, should not get live attenuated vaccines. There's a very, very tiny chance that the virus can mutate and become stronger and more virulent, while people with stronger immune systems, like you and me, could fight it off if that were to happen. People that have compromised immune systems cannot, so they shouldn't get these because it could be dangerous to their health. Inactivated vaccines are also time-consuming to develop. They take years still. There's also still somewhat of a risk, which we'll get to that later, why the risk is no longer really there, but I'm sure some of you remember the Cutter incident with the polio vaccine. In this vaccine, there were live virions, so virus particles inside the vaccines. They weren't killed all the way. So this actually caused an outbreak and it killed 10 children. So recombinant vaccines are cheaper, they're safer, and they're easier and faster to make for scientists. They have less limitations for people like those with diabetes or the elderly or newborns. The disadvantage to recombinant vaccines is that they have short-term effectiveness. So you may have to get booster immunizations due to your immune system not responding as strongly to these recombinant vaccines as they would the other two. So what are recombinant vaccines? Well, unlike the other two vaccines that we talked about that use the entire virus, recombinant vaccines only use part of the virus, usually a protein, and usually a protein that's found on the outside of the virus called a coat protein. So for SARS-CoV-2, the virus that causes COVID-19, we use the spike protein, also referred to as the S protein. It's going to be these guys right here, these little red triangles found on the outside of the virus. But we're not taking these proteins, like these little red triangles, and cutting them off the virus and injecting those into your body. Even better, we're taking just the coding sequence for those proteins. So it's not going to be the actual physical proteins, it's just going to be the mRNA that codes for these proteins. And what we're going to do with that mRNA is put it into a vector. Now, a vector in genetics is just referring to something that kind of acts like a vehicle for DNA or mRNA, which is what this is, to be put into, and then that little vehicle takes it to wherever you're trying to put it into. So in our case, it's going to be our bodies. So this vector is going to take the mRNA, and the vector can be a yeast cell, a lipid nanoparticle, which is what this vaccine is going to use, a plasmid, or other deactivated and harmless viruses such as the vaccinia virus or the adenovirus, both of which are harmless to us, especially when they're deactivated. They just act as a vehicle to carry this mRNA. But like I said, this vaccine is going to be using a lipid nanoparticle. Lipid just means fatty, nano means small, and particle means particle. So it's just like a fatty, tiny particle that's going to take this mRNA. So the vector that has that mRNA is going to be put into a vaccine, and then that vaccine is going to enter your bloodstream, get into your cells, and your cells are going to translate that mRNA and start making the physical proteins, and it's going to start sending these proteins out of your cells. That's going to prompt an immune response and memory, and you'll be immune to COVID-19. So now I'm going to show you a picture representation of what I just said. So here we have the entire genome sequence of SARS-CoV-2. If you were given a little machine and you entered this entire sequence into the machine, a little tiny COVID-19 virus would pop out, a SARS-CoV-2 virus would come out. However, that's not what we're using. Like I said, we're only using a part of the genome sequence. And that part of the genome sequence is going to be the part that codes for the S protein. So scientists took that little segment out, got the S protein mRNA, and then they put the mRNA into a lipid nanoparticle. Those blue things are just like the little fatty things. The lipid nanoparticle, by the way, is completely harmless to you. So the mRNA is put into the lipid nanoparticle, and the lipid nanoparticle is put into a vaccine, which then gets into a human cell. So inside the cell, the mRNA is released, and this is a ribosome that you might remember from one of your biology classes. This is the little machine I was talking about earlier that can take mRNA and turn it into something tangible. So this ribosome is going to process this mRNA, and then non-virulent S proteins are going to come out of that cell. Non-virulent means they're not going to infect you or cause any sort of harm. So then what happens? See this cell that's producing all of these S proteins that the mRNA got into? That's when the immune response happens. So there are these little things in your body called macrophages, and their job is to eat the cells and destroy the cells that are producing these S proteins. But before that happens, your B cells are going to create antibodies, and they're specific to the S protein. So the antibody is what I talked about earlier on, and these are the things that are going to attach to those S proteins, whether they're attached to a virus or not, and tag them for degradation by these guys. So these guys are going to be on the lookout for S proteins, and they know exactly what they look like too. And then your T cells in your immune system are going to remain on the lookout for future S proteins. They have up these wanted signs for the SARS-CoV-2 virus and said, hey, maybe wearing this disguise. So it's going to attack anything that either looks like this or like this. And the thing about the recombinant vaccines that I do want to mention is that this wanted sign isn't as permanent as it would be if it was a live-attenuated vaccine or an inactivated vaccine. Because those other two vaccines prompt a stronger immune response, this wanted sign in those vaccines is going to stay up in your body a lot longer. However, the recombinant vaccines, because it's only using these S proteins, it's not prompting that big of an immune response. So you're probably not going to get a fever or the sniffles or whatever. But the problem with that is you're not going to stay immune to it for as long as you would with the other vaccines. So this wanted sign is just kind of put up with some cheap Scotch tape and it'll fall down eventually. That's where the booster immunizations come into play. So how can recombinant vaccines be improved? Adjuvants. So adjuvants are just things that are added to the vaccine that can strengthen the immune response. They strengthen the virulence, not necessarily the virulence because it's just the protein, but they make more of these S proteins, make them stronger and harder to destroy, which prompts a stronger immune response by you. So then you are a little more likely to have a smaller immune reaction to it. So you'll probably get more things like a fever and feel more tired and everything like that. But it's very important to note that just because you're having an immune response does not mean you're infected with the virus. That is how vaccines work. That's a good thing that you're having an immune response, because that means your body is building everything that we talked about in the last slide to protect you from future viruses. So these adjuvants strengthen them and strengthen your immune response, which means that it's going to lengthen the vaccine's effectiveness, and you're probably not going to have to get booster immunizations every year like you would without the adjuvants added. Another way it could be improved is maybe universal application. So they're doing this right now with the influenza virus. There are specific proteins on the influenza viruses. There's all kinds, but they always use the same protein. It's called hemagglutinin, which attaches to a specific structure on our cells to infect us. So they need that hemagglutinin protein to infect us. So because they all have this hemagglutinin protein, the vaccine is made with said protein and will hopefully be universal to all strains of the flu, A, B, and no matter if it mutates, as long as it doesn't mutate that protein. So we would have to find a common protein located on the outside of coronaviruses, and I haven't looked into this, but S proteins might be universal, and then we could create a universal vaccine for coronaviruses. So now we're going to get into a few anti-vaxxer claims, and I'm going to explain if they're true or false and explain why. So the first anti-vaxxer claim is the vaccine can give you a viral infection. You should know by what we've already discussed that for the live attenuated vaccines, it can maybe kind of sort of be true. So there, like I said at the beginning, there's a really small chance that the weakened virus can become stronger and become more virulent and, have a stronger infection mechanism. However, like I said, normal, healthy people with strong immune systems can fight this off. But this is why immunocompromised people should not receive the live attenuated vaccines. However, for the other two, this is completely false. For inactivated vaccines, like I said with the polio, there have been incidents in the past. But since then, scientists have created inactivation assays to really test these vaccines to really make sure that there are no live viruses inside the vaccine. It is impossible now for active and live viruses to get past these screenings and make it into a vaccine. So these assays are making sure there's complete inactivation. And for recombinant vaccines, we all know that this is completely false because there's no virus, alive or dead, inside of the vaccines. Only the coding for the S proteins. And then even after your body makes the S proteins, they can't do anything bad to your body by itself. It's just the S proteins attach to the ACE2 receptors on your cells, and it's just like the key to get into your locked door being your cells. So the S protein doesn't do anything with infection, and it cannot infect you on its own. Another anti-vaxxer claim would be natural immunity or herd immunity and better hygiene like hand washing are more effective than vaccines. Let's see what the cat has to say about it. False for all three. We'll start with live-attenuated vaccines and why that's false. I'll give an example. Yellow fever. I did some searching online and I found some statistics on it. 20 to 50% of people that get yellow fever, not the vaccine, that get infected with yellow fever die. 20 to 50% of them. So the vaccine has been given to over 500 million people, and only 35 have died after the vaccination. It was never confirmed whether or not the vaccine was the thing that caused their death, but 35 documented deaths post-vaccination have been recorded for over 500 million people. I'm no mathematician, but I don't think that's a very large percentage. Inactivated vaccines, that is false. Given that the vaccine is not for a very deadly virus, it's about equivalent to natural immunity. So you're going to have the same type of immunity that you would from getting natural immunity to it and getting sick with it, and then having immunity. So like when you have chickenpox, you get sick with chickenpox and then you can't get chickenpox again. That's immunity. That's natural immunity. Inactivated vaccines give you about the same immunity. As long as it's not a deadly virus, then getting the vaccine would be better than going through natural immunity. And lastly, recombinant vaccines. This is completely false. Again, there is no virus in these vaccines. So actually, recombinant vaccines could be safer than going through the process of acquiring natural immunity because you don't know the long-term effects that a virus could have on you. So yeah, you went through natural immunity with chickenpox, say, but now you're at risk for getting shingles when you're older. So getting a recombinant vaccine would be a lot safer than going the other route. And of course, my favorite, have fun getting microchipped. There's a group of people out there that believe the COVID-19 virus itself is a conspiracy and that the vaccines are being created with microchips to track people as if they didn't have cell phones that already did that. So an anti-vaxxer claim would be have fun getting microchipped, and that is completely false. I don't really feel like I owe much of an explanation because it's just kind of a silly claim from the anti-vaxxer community. So if there are any questions, comments, threats, concerns, feel free to leave them in the comments or send me a message if you know me personally. I would be really happy to have a discussion with you if you want more information or if you want statistics or readings, I can give them to you. I'm open to having a discussion with anybody about this. So please stay safe. Please get your flu shot this year and consider getting the COVID-19 vaccine. I don't believe it should be mandatory because, you know, freedom in America and all that stuff, but I do believe that if you can, you should get it because that's the only way we're going to fight this and get over it. Stay safe, everyone, and have a happy holiday.